Okay, so this will just walk through it. Remember, the first enzyme that comes along here is topoisomerase, right? And that comes along and it starts making the replication bubble, right? So it just basically lays this thing flat and starts splitting it apart, right? So that's topoisomerase. As soon as there's a little split in here, the two enzymes, or the enzyme helicase, starts shoving its way into what's called the fork. So like where it starts splitting, right? So there's a fork over here. Keep in mind visually there's a fork over here, and there's also going to be a fork on this side then, all right? So topoisomerase starts the split, helicase shoves itself in here and starts unzipping. Where's the weak point in the bonds here? The hydrogen bonds in between, so that's the weakest point, so that's where it unzips. Now if it just unzipped it and then just went along, this would rezip. Why? Because the hydrogen bonds just attract to each other, right? So how do you keep them like pried apart and wedged apart? You use those single strand binding proteins. We talked about that, I hope, right? Did we? Okay. So they just basically just wedge in there, right? And again, I'm doing this kind of quickly. Some of you are recording this, that's fine. Others of you will talk about it later if you need to. But I'm assuming that you've already taken notes. Am I right? Okay.
Yes.
And I apologize if I'm repeating this, but I think it's worth repeating. So these single strand binding proteins wedge this thing apart, keep them from coming back together. Okay? At that point, primase lays down primers, beginning points, right? How many pieces at a time, how many nucleic acid at a time? About 5 to 10, somewhere in there. Not many, but a couple, okay? So they lay down a bunch of little pieces in a row here, and they let them sit in the middle. Primase doesn't lay down DNA. What does it lay down? RNA. Now it uses the same pairing rules, so like T always pairs with A, and A always pairs with T, and G always pairs with C, and C with G. And it goes along. The flaw that will come about here in a little while is that primase can only work in a 5' to 3' direction, right? So when it lays down the new strand goes from 5' in the direction of the 3' end. So the new strand, okay? So if I was to take this apart a little bit more, we'll find out which way primase would go here. You can't always assume, some of you guys when you take your notes, and I watch kids do this on the test too. Oh, by the way, I'm trying to keep unwinding this and this is getting all torqued up over here. What's the enzyme that would come down here and fix this? Topoisomerase keeps going ahead here. A little bit more information on how it does it. It's a really dangerous game it plays. It literally chops the DNA here, like cuts it in half, and lets it unspin, and then glues it back together. It holds it in place. So it actually cuts your DNA into pieces and then glues them back together. It's pretty dangerous. So it doesn't just unwind the thing. It actually like cuts it and makes it so it doesn't have so much stress. Now, if I was to lay this thing out in front of me, I'm going to pull this apart just so we can see this. Let's see what we got here. So this end here is the what end on this guy? That's the 5'. That's the 5' end. So in front of me here, I have from right to left, I have 3' to 5'. Make sense? That means by default, the other side then, from right to left, is 5' to 3'. Make sense? Okay. When primase lays down new stuff, if this end is the 5' end over here on our original strand, our template, what's this strand going to be like right here? On this yellow one, this is going to pair up with a, what color? With a green. Oops, here's green. There we go. Primase is going to lay green with that guy. And which end is going to be pointing which way? So this is 5' over here. So it's going to lay this guy down so that 5' points that way. You guys get that? Okay. And then it's going to continue in a 5' to 3' direction. So which way is primase going to move here? This way or that way? The 5' end is over here. We're going to go that way. 5', 3'. So primase is actually going to work this way on this side. You have to check.
So when primase works, it goes from 3 to 5?
It works from 5 to 3 on its own side. On its own side. So it's going to work like this. This is 5' over here. And then it's going to work in a 3' direction. So it's going to keep going this way. Okay. So it's going to lay down a couple of primers. Then DNA pull 3 is going to come in here, and it's going to find that primer, that starting point. It's going to go, okay, I got to pick it up here, and it starts moving towards a replication fork. It can just move indefinitely, like unlimited, right? As long as this thing keeps doing what? Splitting and spreading apart. As long as the helicase, the topoisomerase, and those single-stranded binding proteins keep wedging things apart, as long as that keeps going, this guy can just keep, it can stay locked onto the DNA and just go and go and go and go. All the way until it hits the what? The end. All the way until it hits the very end of this thing. Just keeps going on this side. Now over here, back here, can polymerase just flip and go the other direction? No, because it always goes 5' to 3'. So what has to happen on the other side? Visually, this is what happens, and I don't know how to describe this other than just do this visually. Remember, primase can only lay this way on this strand. So what primase does is it hops back to like the replication fork on this side, and it lays a primer over here, and then pull 3 hops on, and it goes until it hits the next primer. And then it hops off. So we lay a little segment in between there, in between the primers. Unwinds, primer moves back another step. Polymerase moves back, moves forward. Back a step, forward a step, back a step, forward a step. Does that make sense? So ultimately we get little segments in here. Did I tell you what those segments are called? I hope I did. Did I tell you what those little segments are called? These little segments have names. You need to know the name. They're called, and you can go back to your tables when you do that, but they're called Okazaki fragments. Okazaki fragments. They're spelled just how they sound. I know it sounds like a weird word, but it's not hard to spell. Okazaki fragments. Sound it out, Paige. Come on. Okazaki fragments. Okay. Yep. Okazaki fragments. Those are the little pieces that are made, basically, like, I'll call it backwards order. I don't know what you want to call it. Like, they're the little tiny segments that are made on which strand? Leading strand or lagging strand? On the lagging strand. So on this lagging strand. Okay? Now, notice, and this is the one that if I want to test to see if you understand this, here's what I'll do. I'll label this right here, 5'. Let me think here. I'll say, okay, so primase starts here and it moves in a 5' to 3' direction. It's going to go that way. What does that make this right here? Leading or lagging? Leading, because it's going towards the replication fork. That makes this back here lagging. What does that make this side? It's kind of a trick question. What does that make this side?
Lag.
It's both. Because now primase, when it's working on this side, will work in which direction? That way. So what is this side right now? Leading, and then this would be the lagging strand back here, because you'd have to keep unzipping to back primase up. Keep unzipping back primase up. So each side has its own leading and lagging segments. Does that make sense? So many times you guys want to compartmentalize. You want to go leading, lagging. You want to do that in your head. You want to say the top. Okay, the top is leading, the bottom's lagging. No, you got to look at it. You have to look at it. Okay. Now, so polymerase 3 lays down its little segment. Okay, it gets done. What does pull 1 do? It comes back and it spell checks, right? You cover that? And it replaces what pieces? Did we get to that? No? No. Okay. So DNA pull 1 goes back and spell checks the primers, and it replaces all the RNA with DNA. So it puts the right stuff in. So DNA pull 1 goes back to every primer, and it replaces all the RNA with DNA. Can somebody close that door? I'll wait till the door is closed, because I honestly won't do it. Thanks, Isaac.
Got this.
All right, so, question.
So, polymerase 3 comes in and finishes
It fills it, it finds primers, it finds starting points, and it just starts there, and it just goes, and it goes. It places in the correct DNAs. The only problems that once this is all done, the only problem is that the primase has put in RNA instead of DNA, And that's what pull one does. It goes back and replaces all of those. Okay? Now, pull one is kind of a dumb little molecule in that when it replaces the RNAs, it doesn't glue the two pieces together from where pull three put down its nucleic acids and pull one stopped. So there's always a gap there. So the final enzyme that comes along is called ligase, and ligase glues all the pieces together. If we wanted to get a real idea of what that would look like, there we go. So here's primase. It's going to work in a 5' to 3' direction. So this is the 5' end over there, right? So which one's going to go next, this guy or this guy? If 5' is over this way, 3' that way, which one is it going to go to next? This one. It's going to go to this one, right? So it's going to go to this one. Primase is going to put this guy down now. It's going to pair that guy up, and then it's going to put these guys on there. Uh-oh. Get lined up right. So let's say primase puts those two guys down. So that's the primer, right? Let's assume that's the primer. I'm not going to do all 5 to 10 of them, so let's just assume it's just 2. Then pull 3 comes along and goes, hey, a starting point. And it'll start from here and keep going that direction. So it's going to put in a blue one, an orange one, right? What's next? Green, right? So it's going to put a green one in here. This guy's going to go here. And it'll put all this together, right? So it starts putting this together. Now, when pull 1 comes back, it replaces these guys with the right nucleic acids, but it leaves a gap between these guys. I'm leaving this kind of sloppy for you, sorry. There we go. So pull 3 went along here and put all this stuff in here. And then pull 1 is going to replace these guys and put the right things in, but it's going to leave this gap so the DNA isn't stuck together. Ligase is going to come in here and glue these two pieces together. It's going to make them work. Some of you are looking at this visually, realize I've flipped this, but that's okay. All right, just realize that ligase glues the pieces together. Okay? Make sense? At least a little bit? Yeah.